Good morning. It is a total pleasure to introduce Steve Stearns, the first speaker of the day. I think one of the hardest things in science is to figure out when you should be gentle on an idea, when the idea or the person needs nurturing, and when you should be rigorous, when you should hit the person or the idea with as much rigor, scientific rigor as you can. And finding this balance between being gentle on ideas and injecting rigor is, I think, one of the trickiest things in this business. That's true of ideas and people. And Steve Stearns has, since the 1970s, been doing this in a number of different fields. He practically constructed the whole area of life history evolution. He practically constructed European evolutionary biology, and if you doubt that, talk to somebody who was in Europe in the 1980s at the state of the play. Steve developed a field by mentoring people and developing a journal and a fantastically successful meeting series. He built evolutionary biology in Europe. And then, of course, in our field of evolution and medicine, he's been a seminal player right from the get-go. He started building our field with the edited volume, or two edited volumes now, Evolution and Health and Disease, first starting with the Sion meeting way back in the early 90s, I think. 96, okay, there you go, the mid-90s. He's gone on, of course, to write many important things in the area, not least his work on whether humans are still evolving and how they are still evolving. He's just finishing an evolutionary medicine textbook with Ruslan Metsisov, which I'm sure we're all looking forward to. And of course, most important of all, Steve is the founding editor of our, and I say that advisedly, our journal. And the issue with the journal in the early stages is that you need to develop, obviously, material in the journal, but at the same time, and particularly in this field, you need to make sure that the rigorous standards are upheld. And I think Steve is doing an absolutely marvelous job of balancing this need for nurturing ideas in our business at the same time as injecting rigor. So it's with very great pleasure to introduce Steve now to talk about what is a patient. Thanks, Steve.
Thanks for the kind introduction, and thanks to Randy and his wonderful team for organizing the meeting. Now, one of the things I've learned in science and in teaching is that simple questions are incredibly valuable. This is a simple question. What is a patient? And you're going to get my answer to it today, and my answer, of course, is just the best I could do at the time, and maybe it's okay and maybe it's not, and you all will have your own answers. And I would like to emphasize at the outset that I would defend the importance of this question far beyond what I would defend any of the answers that I would give to it today. I think the answers that I'm going to give are important. I think you'll see that people have already brought up a number of these answers in the course of their talks, but I think that it's questions like this that stick with us. We used to have a comprehensive exam at Yale when you finished your undergraduate degree. It was 2 days. It consisted of 8 questions that you answered. Each one you had an hour to do, so it was 8 hours worth of examinations. And those questions have actually stayed with me over the last 48 years. So I think this would be a good question for a medical student to have to write as part of a comprehensive exam taken at the end of 4 years. Sit down for an hour and just write on what is a patient. Now, the standard course in medical school has been to build up a picture of a patient as a genetic and cellular and biochemical process with lots of points for well-informed interventions, and I'm just taking that as a given. That's all good stuff. What I would like to do today is talk about the consequences of the fact that patients have evolved. Now, this is part of a book that Ruslan and I are writing, to which Andrew just referred. And I'm giving you all the chapters here. So what is a patient is chapter 2, and what is a disease is chapter 3. And you're going to get what is a patient at 8 o'clock in the morning and what is a disease at 1 o'clock in the afternoon from Ruslan. Okay? The others are on defenses, pathogen evolution, cancer, reproductive medicine, mismatch, mental disorders, and then all of the issues that arise in individual versus population health, which are mostly about public goods problems. And I'd like to start off by just having you consider this oncologist, okay? So this is a clinician. He's looking at a woman who has cancer, and he is going to try to treat her with all of the wisdom and empathy and technology and technique that he has available to him. And all of that mental structure has been built up to create an image in his mind of what a patient is, where they came from, and how they work. So, that patients have evolved, remember I'm assuming that all the stuff we know about reductionist medicine is great. I'm just building on that. So, that patients have evolved means at least this. They're bundles of trade-offs. I'll illustrate that with a few. They contain ancient constraints. That has an important message that perhaps some of the billionaires in Silicon Valley haven't fully digested yet. They vary for genetic and environmental reasons that leads us on into personalized medicine. They age, which makes them vulnerable, and Baba explained that to us yesterday, and I will do it again. And they are pretty highly unusual among primates. They also contain very important microbiota, so you can see that many of these things have already been mentioned. But what I want to do is put them together. So here's the picture of the 6 blind men and the elephant. And I want you to go back for a moment and think about that clinician looking at that patient and how we assemble a set of interacting ideas and concepts that give us handles on how to deal with a problem. And the old story of the 6 blind men and the elephant is quite relevant because we have to put them all together in order to understand the elephant. So to begin, trade-offs are everywhere. They're the core of life history theory, but they're also involved in a lot of other stuff. I didn't really realize this until I started talking with Ruslan a lot. Ruslan's an immunobiologist. And Ruslan has taken the life history concepts of trade-offs and cost-benefit analysis and applied them to the immune system and what a disease is and what defenses are and things like that in ways that made me realize that they're just pervasive in the medical context. We'll see a few. So certainly the central one is the one that George Williams emphasized. That's the trade-off between reproduction and survival. I'll give you an example from Framingham. I'd also like to mention that there are trade-offs between mating and immune defense, mediated by testosterone, so macho makes you sick. Mike Muhlenbein has worked on that. Between contraception and breast cancer, Beverly Strassman has emphasized that. By the way, just in case any of you are worried about that, in fact, taking contraceptive pills is probably pretty much a wash because although that increased the risk of breast cancer, it's protective against ovarian cancer. Then there's the whole issue of treating infections and disrupting microbiota, the problem of C. difficile infections. And this list is very, very long. So the trade-off concept is central. And it's something that every physician should understand in looking at a patient because it means you can't do one thing without having side effects. All treatments will have side effects. So here's a trade-off. These are the costs of reproduction in Framingham. I'll walk you through a bit of this. This is the number of women who were measured. So you'll notice that that's 1,000 up here. So there were a bit over 1,000 women in this analysis that had 2 babies. This is how many babies they had per lifetime. And this is the relative mortality risk over here. And so what you can see is that having 1 is better than none and having 2 is better than the first, but the 3rd, 4th, 5th, and so forth are the ones that are causing heightened risk of death. These costs are both phenotypic and genetic. We had the pedigrees in Framingham, so we could analyze that. Women with more children had shorter lives, and each additional child after the second cost about 3/4 of a year of life. More children implied higher risk of death from heart attack, stroke, or cancer in both women and in men. And as the grandfather of a 2.5-year-old, I can understand why. The genetic correlation of family size with age at death was minus 0.69. So that means that there was a genetic basis to this impact of reproduction on lifespan. And in this analysis, we were able to take quite a few cultural effects into account. So that's as you saw yesterday, the literature on this has quite a few papers, and some of them find a genetic cost and some of them don't. And we know from work on Drosophila that you'll only find a cost of reproduction if the organism is somewhat stressed. So, in fact, what we found is that these costs existed in women who had been born up to about 1950 and then disappeared thereafter. So that's point one, trade-offs. Patients are bundles of trade-offs. Point two is that all organisms contain ancient constraints, and these have implications for medicine. Asymmetric cell division is a precondition for aging, and it's a little hard for you to see this diagram up here. Basically what it shows is that in E. coli, the cells that have the new parts have much better survival and reproduce and grow faster than the cells that have old parts. Even E. coli ages. So the condition for what will age and what will not age isn't the germline soma distinction, it's the asymmetric cell division distinction. And it turns out that there aren't any cells that divide symmetrically. Everything ages, and I'd be happy to talk later if you think Hydra is an exception. Secondly, and this picture is Volvox, is to remind me what Carlo Meli has told us all, that very early when we became multicellular, we evolved stem cells for tissue repair, and they predispose us to cancer because they retain the ability to both multiply and differentiate for a very long time. Then we have invasive placentas. It turns out that invasive placentas are ancestral in mammals, and they have been reduced. The invasiveness has been reduced in certain mammals, such as horses and cows, and it's been heightened in others, such as our primate lineage, and they predispose to metastasis. So that's just a little figure of a placenta there. Harvey has much better pictures. And then, of course, there's bipedal locomotion, which complicates birth. So it's hard to see this, but this is a chimpanzee down here, and this is a human over here. And Wenda is going to talk more about this kind of thing. But the whole point of this is that there is a macroevolutionary context in which the patient sits. And there are consequences that go back as far as the origin of life that are important for aging, that go back to the origin of multicellularity, about 1.5 billion years probably, that are important for our vulnerability to cancer, that go back in our hominid lineage and in mammals, two events, probably one at about 15 million and one at about 65 million years, that predispose us to metastatic cancer. And then, of course, bipedal locomotion, it's about 4 or 5 million years old, that complicates birth. So there are ancient constraints. And if a Silicon billionaire says, I want to set up an aging institute which is going to make me immortal, I would like him to reflect on the fact that since the beginning of life we've had asymmetric cell division, and that has set up a difference between new and old parts in daughter cells, and we're not going to get away from that. So a little bit on stem cells. They're a great innovation. They retain the potential to differentiate. That pre-adapts them to a cancerous lifestyle. They can differentiate and they can move. They're all over the body. They're positioned to replace cells that wear out and are discarded, and particularly in active areas like bone marrow, lungs, intestine, and skin. And the embryonic stem cells that invade the endometrium to form the placenta share many traits with metastatic cells. That means that since all the cells in our body contain the complete genetic program to make a whole individual, they also contain that genetic program we used when we were implanting in the endometrium. That means potentially every cell in our body, if the right switches get flicked, can start behaving like a metastatic cancer cell. So one way to look at this is comparatively. This is Alaric de Souza, and this is Gunther Wagner, student and colleague at Yale. And Alaric got interested in this kind of stuff, and with Gunther's help put together this literature survey, which shows that if you compare the rates of skin, glandular epithelium, and connective tissue cancer in cows and horses versus dogs and cats, that the percentage of malignant tumors is much lower in the non-invasive placenta species than it is in the invasive placenta species. So, you know, this kind of logical criterion is just consistency, it's not causality, but it indicates that there may be something to this idea, and it doesn't work for non-glandular epithelium. So there's something interesting going on there. Okay, third point. Patients vary genetically. And this one is actually difficult to communicate to the general public simply because the numbers are so large. After all, if we have 3.3 billion nucleotides in our genome, and I differ from you by 0.1%, I'm still differing from you by 3 million nucleotides. So those big numbers are kind of hard for us to hold in our heads. Patients vary genetically in ability to resist disease. Some of the biggies are malaria, leprosy, AIDS, and dengue. Here's an evocative photo of a malaria GWAS. These Manhattan plots are just all over the place in the literature now. Patients vary in their ability to metabolize drugs. The two big families of enzymes that do this, cytochrome P450s and acetyltransferases, both evolved to deal with toxic plant compounds and have been elaborated in evolution to deal with a huge variety of things. This is the structure of a cytochrome P450. And if we look at humans across the world, we see that there's a tremendous amount of genetic variation. In this picture, which is from the famous Lee et al. Article in Science, the y-axis is 650,000 SNPs, and the x-axis is 938 different human individuals from populations all around the world. And the colors there are showing sort of, with a structure analysis, are showing the way that a clustering algorithm can group origin. So you see that people in Africa are pretty homogeneous at that level of structure. In Europe, fairly homogeneous. In East Asia, fairly homogeneous, and very mixed in the Middle East. A fair amount of mixture in South Central Asia, and so forth. Now, if you take that and you make a phylogenetic tree out of it, it gives extremely strong confirmation of the idea that we all came out of Africa. We're all Africans, that we did so probably about 100,000 years ago, and that we spread out across the planet approximately in this sequence. So down here we have people in the Middle East, and then we go up through some people in South Asia. These are people in China, and then these are Native Americans and Polynesians up here. It's very strong evidence that that was the sequence in which we came out. Now, as we moved across the planet, we encountered different diseases and different foods, and there was genetic drift and founder effects, and we differentiated quite a bit. The result of that is that there's standing variation now in humans for disease resistance. These are the big six of human infectious disease, where genetics is important in resistance to human diseases. So sickle for falciparum, Duffy for vivax malaria. There's a gene that reduces risk of getting prion disease. There is another gene in Melanesia against falciparum, CCR5 against HIV, and a blood group non-secretion gene against Norwalk virus diarrhea. This is all more or less classical genetics candidate gene approaches. This is GWAS discoveries down here. So additional information on HIV, Hep B, dengue, TB, leprosy, meningococcal disease, and again another prion disease. So we're finding a fair amount of information. By the way, the ones up here are all causing differences in odds ratios and risks of more than 3 if you have the homozygote, the mutant homozygote. These are, the GWAS discoveries don't usually have that explanatory power. And there are also single gene associations for some others. The interesting thing to me is that there are about 1,300 or 1,400 human infectious diseases, And the number that pop up where there's significant evidence of resistance is always a much shorter list. So what that means is that there's a huge amount of disease, infectious disease, that physicians will encounter where it's going to be hard to find very much powerful explanation in terms of genetic variation. But of course, in these, there is some. Now, just to give you some example of what this means for drug metabolism, consider cytochrome 2D6, P450 2D6. It can vary from 2 to 13 copies per individual. So people have very different processing capacities for this particular gene product. It metabolizes a quarter of the drugs that are put on the market. The polymorphisms are influencing about 12 to 14% of drug metabolism, including antidepressants, antiarrhythmics, anti-cancer drugs, and analgesics. Duplications are reducing efficiency of drugs that treat arrhythmia, Alzheimer's, and heroin addiction, but they do improve tamoxifen treatment of breast cancer. And as a result of these highly variable individual reactions, the drug industry now avoids making drugs that are metabolized by this cytochrome P450 because it is so difficult to do the risk analysis. There's a very important message in the other big class of drug metabolizing enzymes that are variable genetically in human populations. These are the N-acetyltransferases. I just want to pick out how they mediate cancer risk because it carries the message that genotype by environment interactions are everywhere. So for bladder cancer, those that have a combination of a slow acetylator at one and a fast acetylator at another, and who have ever smoked cigarettes. So the genetic variation is in which version you've got of those two loci, and the environmental component is whether you smoke or not. That group was 2.73 times as likely to get bladder cancer. Or consider colorectal cancer. The fast acetylators had significantly higher risk of colorectal cancer than the slow ones. Smoking intensity increased colorectal cancer risk among carriers of both NAT and NAT1 and NA2 fast alleles. So again, the environmental component, smoking or not, is interacting with the genetic variation. Pancreatic cancer. So the NAT1 polymorphism is interacting with a dietary mutagen to increase the risk of pancreatic cancer in Japan about 2 to 2.5 times, but it's doing it in men but not in women. Okay, so these things are all complex gene, in this case, gene by environment by gender interactions. Next point, patients vary environmentally. They are points on reaction norms. So Joe Alcock brought this up. This is something that Peter Gluckman's group has contributed to massively. If you're born thin, you're at increased risk of metabolic syndrome. And I'll come to a little bit more info on that in a minute. If you're born by C-section, you're at increased risk of allergy, asthma, and obesity. If you are breastfed, you have less risk of allergy, obesity, and you have better immune protection. And if you have multiple antibiotic treatments in childhood, you're at increased risk of obesity and allergies. Many of these things are mediated by the microbiome. So here's a good example of what a reaction norm looks like in humans, okay? These guys are identical twins. This is Evald and Otto Spitz. Evald on the left ran distance, and, Otto ran distance and Evald lifted weights starting at age 18. So this is 4 years later. They're identical twins. They have precisely the same genetic composition. So, sometimes a picture is worth 1,000 words. You can tell people that your point's on a reaction norm. Well, these guys are two different points on the same genetic reaction norm. Now, let's go to a medical consequence of that fact. And this is the data that kicked off the DOHAD boom, the developmental origins of health and disease. This is what happened in the Dutch hunger winter when the Nazis blockaded food shipments into the Netherlands, starting in about November of 1944 and ending in about April of 1945. During that period, women were in different stages of pregnancy. They were either late, middle, or early in pregnancy when they hit that famine. They were trying to make it on about 700 or 800 calories a day. Here are the consequences decades later. This is a horrible big table, so I'll just summarize the take-home points in it. When that stress occurred mattered to which kind of disease an adult developed 40, 50, 60 years later. Early gestation, then you get cardiovascular disease and general poor health. If it was in mid-gestation, it was kidney disease associated with diabetes and lung disease, and in late gestation, it was insulin resistance leading to type 2 diabetes and metabolic syndrome. So that set off the whole cascade of insights into the idea that things that are important very early in life affect health late in life, and that's been hugely developed since then. Now, of course, patients also evolved to age, and that accounts for our vulnerabilities to degenerative disease, cardiovascular disease, cancer, neurodegeneration, Alzheimer's, things like that. The proximal causes of aging differ among individuals. Each of us breaks down for unique combinations of reasons. Now, of course, there are themes in here, and some things are more frequent. Interestingly, they change with time, so that while many more people died earlier of cardiovascular disease in the developed world, now that is shifting much more to cancer and neurodegeneration. And if we fix one problem, another one will emerge. That's because we are complex, we have many parts, and we break down differentially and individually in those different parts. So I'm going to give you the evolutionary theory of aging in one slide. Baba gave it to you in one rap tune. Peter Medawar observed that the force of natural selection weakens. This is basically the statement that, hey, evolution is a numbers game. And if we just think about how old our mothers were and our grandmothers were and so forth when they gave birth, we can see what age class was contributing genes to the next generation. And Medawar's point is that even in a theoretically immortal population, this will happen. The older ones will be under less selection simply because there are fewer of them. They will be dying, even if they're potentially immortal, they'll be dying for extrinsic reasons, accidents, predators, things like that, then so the older ones have much less selection on them than the younger ones. So even if organisms don't age, there are going to be fewer old than young. Now, George Williams then contributed the important insight that if a gene has positive effects early in life, they will often more than compensate for any negative effects late in life. That's the antagonistic pleiotropy thing that Baba was having a hard time saying, the rap song. This declining force of selection combines with antagonistic pleiotropy to evolve organisms in which aging is a byproduct of reproductive performance. Aging is not selected directly. There isn't any natural selection to eliminate old things from the population to make room for young things. What's going on is that natural selection is trying to optimize reproductive performance. It does so most effectively by operating on the young, and the byproduct is that the old fall apart. So does it happen in humans? Well, is there antagonistic pleiotropy in humans? There's some evidence. Genes have now been found that are associated with increased fertility early in life that increase cancer late in life. One of them is P53. So it is a very famous cancer susceptibility gene. Of course, there aren't really any genes for cancer. These are all genes that do other important things, and when they mutate, they increase risk of cancer because that other important function isn't being done anymore. So P53 is a gene that controls cell cycle and DNA repair and things like that. And they reduce the risk, the mutants that reduce risk of implantation failure increase risk of cancer. There's nothing earlier in life than implantation. You get a mutant in this gene as a little zygote, and you're going to go dive into that endometrium and implant in it. If you've got the right variant, you're going to do better. And gosh, you don't have to pay the risk of cancer until 5 or 6 decades later. Another famous cancer gene, BRCA. Alleles of BRCA that increase risk of breast cancer are associated with larger families, reduced birth interval, and a longer period of childbearing. I think this one needs to be confirmed. I think that that one needs to be confirmed too. But what I'm saying is that we have suggestive evidence for antagonistic pleiotropy in humans. A third example is a gene called EOMES. It's kind of a master control gene. Variants of this are associated with risk of bladder cancer, multiple sclerosis, and also with larger families. So it looks like these trade-offs, the genetic word for trade-off is antagonistic pleiotropy. That's one level at which a trade-off can be analyzed. Look to be real in humans, and they carry the somewhat discouraging message, if you're older, that stuff that was good for you early in life is going to get you late in life. Now, humans are also unusual primates. So I'm now going to talk about things that have happened to us mostly since we shared ancestors with chimpanzees. We have large, fat, relatively undeveloped infants. David Haig taught me that our babies are actually fatter than baby elephant seals. We wean them early, and we have short interbirth intervals, which requires actually a social support structure. Our infants have slow development and extended childhood, which is a whole life history stage not shared with other organisms. We usually have one offspring. We occasionally have two. And women have menopause. We all have a much longer life than our closest relatives. So now I'd like to thank David Haig. I've been using this slide for some time. Thank you. It's a tale of two mothers, Emma and Fifi. So Emma had 10 children in 17 years. 7 survived. Her interbirth interval was 1.8 years. Age at first birth, 31. Age at last birth, 48. She lived to be 88 years old. Fifi had 9 children in 31 years. 7 survived, same number. Her interbirth interval was 3.9 years. Her age at first birth was 13 years. Her age at last birth was 44, and her age at death was 46. Emma was Charles Darwin's wife, and Fifi is a chimpanzee. Now, if we compare, the dotted lines here are survival curves. The dark black lines are fertility curves. If we compare the fertility curves of chimpanzees, hunter-gatherers, what we find is that humans differ from chimpanzees not in their fertility curves, which if we line them up are just about on top of each other. They differ in how long they live after menopause. About 20 years, and now even more. So why menopause? This one is actually a wonderful discussion thing for seminars, and it makes a great critical topic for papers because students really have to think for themselves because we don't know why, okay? So I'm going to give you the hypotheses. The first is the mother hypothesis, which is that menopause is there because it's a terminal reproductive investment. In other words, the mother stops reproducing when her body, quote, knows that she won't be around anymore to raise that child, and she's got to stick around a while because humans have extended parental care. Then there's the grandmother hypothesis. So that is the idea that we survive after menopause to better help daughters rear grandchildren. That's George Williams and Kristen Hawk's hypothesis. Then there's the byproduct hypothesis. I am one of the people who advanced it. I'm sure there's plenty more. Women have menopause because they run out of oocytes because they're screening for quality. Okay? So it's more mechanistic, and that then focuses you more on the early advantages of screening for quality rather than the late disadvantages of not having to produce children anymore. And then there's Peter Ellison's self-domestication hypothesis, which is that we domesticated ourselves, so we're kind of like zoo animals. We have greatly lowered the extrinsic mortality rate, which is why you saw those survival curves for humans going out much longer than chimpanzees. We probably do that because of our improved social structure and niche construction and culture. And evolution said basically, well, as long as these people are hanging around after the point at which they used to die, we might as well make some use of them. and therefore evolve transgenerational transfers like grandmothers and grandfathers give to grandchildren, and so forth. So that's Peter's hypothesis. We don't know. And actually, the truth is probably a mix of some of these, but it's been hard to sort out. Now, my final point is that patients are largely microbiota. This is well known now. About, and as Katie said yesterday, these numbers change, but we are about 10 trillion mammal cells and about 90 trillion cells of bacteria and worms and fungi, and then lots more viruses than that. This co-evolved microbiota is protecting patients from infections. It digests certain foods, provides essential nutrients, and emits signals that both initiate the proper development of the immune system and maintain its health. So we're in continual crosstalk with our microbiota. Many of the diseases of civilization result from disrupting microbiota function. One example has to do with antibiotics, Clostridium difficile, and fecal transplants. Nosocomial, that means hospital-acquired Clostridium difficile infections, result from antibiotic use for anything. I mean, you might go in and you need antibiotics for surgery or for some other thing, but it disrupts your intestinal microbiota, whacks down the competitors of Clostridium. It takes off and causes terrible diarrhea, causes about 14,000 deaths annually in the US and costs more than $1 billion a year to treat. The antibiotics that are used to treat it include these three, but there are multiply resistant strains out there now because these antibiotics have been used so much. And there's a nice paper just from last fall from a group at Harvard, frozen fecal inoculi, so this is fecal transplant therapy, from unrelated donors. So this isn't like not getting it from your spouse, just from, a hospital's got a fecal transplant bank in the fridge. Cured 90% of the patients that were suffering from multiply resistant C. difficile infections, and they restored the natural gut ecosystem. So this is the kind of practical treatment that can result from recognizing, hey, we co-evolved with these things, and when we mess them up, we've got to get them healthy again. Okay? So my conclusions are that because patients are bundles of trade-offs, most interventions have side effects, some of which are important, and we need to be aware of them. Think back to that picture of the doctor looking at the patient, okay? Needs that kind of thing in his mind. Because some diseases are byproducts of ancient constraints, such as cancer, they can be treated but not eliminated. You will find statements out there from things like our cancer center in New Haven that we're going to win the war on cancer and we will become free of it at last. We're never going to get away from cancer. It is an inevitable byproduct of the way we're constructed. We shouldn't be raising unrealistic hopes while we treat as well as we can. Because patients vary genetically in disease resistance and drug response, it is true that individualized medicine can help somewhat. It's a bit limited by the power of GWAS, which is usually low, but it can help somewhat. I think President Obama has oversold himself on this one, but it can help. Because patients are points on reaction norms, in part produced by events early in life, we need medical histories of the entire life, and we need to think of the entire life when we are looking at any of the conditions that arise because of early environmental experience. And because patients contain huge populations of commensal microbiota that have key functions, we should be very careful about disrupting them. Thank you very much.
Thanks very much, Steve. Excellent overview. We've got time for a few questions if people have questions or comments. Kathy, do we have microphones? Yeah, thanks.
Does Neil ever not ask a question? Neil?
No, we need to follow the microphones.
Oh, okay.
Yeah, I just wanted to quibble a little bit about the asymmetrical division, cell division versus the germ-soma distinction as the requirement for the evolution of senescence. I've published a couple of things on this where I've argued that, so if you look at Stewart's E. coli, you look at the two daughter cells before the division occurs, so proto-daughter cells, I guess. What I think is happening is the one that goes on to senesce, by not devoting enough resources to repair or maintain it to get it up to par with the other daughter cell that doesn't go on to senesce, I see that as a reproductive transaction. I see it as nepotism, really. So that what happens is by foregoing that investment, the other daughter cell gains, and Stuart showed that, and you showed it in your slide, you get an increased chance of survival and reproduction. So that reproduction that occurs, or through nepotism, has that old pole cell, as it finally emerges as a daughter cell, has already reproduced. The force of selection is already weaker, acting on it, and so that's age structure that allows senescence to creep in. It's also an antagonistic pleiotropic transaction, I think. I won't dwell on that, but so we ask ourselves, what's a soma? Well, a soma is a collection of molecules and structures that supports what's going to be the germline, keeps it alive, gets it into their next generation. And what's a, sorry about that.
Hey, Paul, you're giving a whole lecture. You're not asking a question.
Okay, so that is a little bit soma, I think. So I would argue we want to call it a germ-soma asymmetry.
Doesn't bother me at all. What you're doing is you're switching the labels, but the underlying process remains. Fine with me.
Neil.
Yeah, so I applaud your skepticism with respect to the claims of cancer centers and certain others who emphasize buzzwords like precision medicine, and essentially what the cancer centers claim often meet the criteria for Harry Frankfurt's definition of BS. So a couple other points. One is...
I think there are several people around the question, so we could maybe get a reaction from Steve on that one.
Okay.
Yes. Next.
Could you pass the mic to Mark, please?
Oh, we got a mic over here. Is that right?
Okay.
Is it on? It's on. Hi, Margaret Humphreys, Duke University. Thinking about your menopause slide, I sort of wondered if all those theories were postulated by males. Maybe that's unfair. But it made me wonder...
Last I looked, Kristen Hawkes had 2 X's.
Oh, okay. All right. So I don't know all these people. Thinking about menopause against the cost of a female being fertile, the horror of monthly menstruation for humans compared to, I think, other primates, other animals, versus the release of being done with that and all just the bodily cost of menstruation. If you look at the question that way, rather than the end of fertility, which of course is part of it, if that changes your perspective at all.
Well, there are quite a few species that menstruate that are non-human.
Oh, I know that, but dogs menstruate all right, but you barely notice it, at least the dogs I've known, compared to the heavy, painful, difficult menstrual flow of humans that I dealt with as an internist.
There are certainly going to be lots of costs with being reproductively capable in humans, and there's no question that that's an important issue, and there's a whole literature on the evolution of menstruation as well, and lots of competing hypotheses about that one as well. And yes, it must certainly interact and contribute to the costs and benefits of menopause, no doubt about it. But whether or not the cost of menstruation is going to be large enough to counterbalance the benefit of an additional child in an evolutionary sense seems to me to be a little unlikely. In other words, the cost will be real, but I doubt it will be a decisive contributor to that particular argument.
Okay, we have time for maybe 2 more questions. Mark, and then there was one down here. Yeah, Kathy, just here.
Hi, Steve. Mark Schwartz from NYU. Whenever I listen to you, Steve, I get curiouser and curiouser. And as a physician, I think about this message being conveyed to physicians and scientists, and I begin wondering, well, what about, how would patients hear this? And then I realize, wait a minute, we've got a room full of patients here. And I'm curious about the potential impact of this messaging on our illness behavior, which is the way we behave when we're sick. And it's just an interesting hypothesis to think about testing of what impact this sort of messaging to the general public might have on patients' illness behavior. Just a thought.
That's an interesting comment because we now have at least 2 hypotheses about the therapeutic value of evolutionary explanation. One is the observation by clinicians that patients prefer to get the why it evolved kind of explanation of their condition rather than the reductionist explanation. It makes them feel better. But now we have this new idea from Mark that it might actually cause patients to throw up their hands and say, oh, it evolved, so I can't get treated, which would be a mistake. I'm taking kind of an extreme form of your hypothesis, but it's useful to play them off against each other. That's productive. That could go somewhere.
Okay, last question.
Phil Askinase, Yale. A lot of what you said was biological, but it's said that we're the first species to evolve in our culture. Do you think there's something to that?
Thank you, Phil. It all depends on what one means by culture. There is culture in chimpanzees, but I doubt that it's contributing quite as much to their health and disease as the niche construction of our massive human biomedical establishment has contributed to our current health state. Chimpanzees do seek out medicinally effective plants when they're sick, so they do medicate a bit. They have a bit of medical culture, but it's nothing like what we have in humans. So yes, there are adumbrations of this kind of thing in other species, but nothing to the scale that it exists in humans.
Okay, we have to stop there. Thanks very much, Steve, for a very thought-provoking talk.